Mobile elements can shape genomes basically in two ways. First, when they move from one position to a new one, but also because the presence of thousands of copies of very similar fragments of DNA scattered throughout the genome can lead to different types of rearrangements if they recombine. Let's see each of these two mechanisms in more detail. When mobile elements actually move in the germline, they can disrupt or change existing genes. This happens very seldom, of course. In humans, a new insertion of an ALU element happens once every 200 births, according to the latest data that we have. But over long evolutionary periods, this can add up to a substantial amount of genomic changes. The most common disruptive mechanism is insertional inactivation of a gene. The element interrupts the coding potential of the gene, breaking the reading frame, for instance. But sometimes a mobile element carries with it an exon which is nearby, and it inserts itself together with that exon in a different gene. The upshot being that this gene now has a new exon so that it codes for a new isoform of the protein with a few more amino acids or perhaps a new protein domain. In other cases, it is the mobile element itself which is partially exonized. That is, the element moves to the intron of a host gene, and then part of it is recognized as a new exon because it contains the sequences, the right consensus sequences for splicing. So the protein encoded by that gene will also change. So it is easy to see how all these processes will create new combinations of genetic modules and also lead to several proteins sharing the same domains. Then there is a second mechanism. As I said, the ubiquitous presence of these highly similar elements dispersed throughout the genome poses another threat, the threat of illegitimate recombination during meiosis, that is, recombination between sequences which are in different locations in the genome so that they should not recombine in normal circumstances. This process, known as unequal crossing over, results in the duplication or deletion of genomic segments. Evolutionary, duplications are more important than deletions in terms of creating new variants. Deletions will disrupt the gene or lead to a shorter protein, but duplications have more potential for increasing the functional complexity of a genome. For instance, unequal crossing over between mobile elements flanking an exon can lead to the duplication of that exon. And this is thought to be the origin of repeats of exons which are common in some genes. You see the same exon repeated several times in tandem. And this, in the corresponding protein, leads to repeats of amino acids or repeats of protein domains. And if it's not an exon but a whole gene that is duplicated, things get much more interesting.